yet another biology revision stream. It is not long now until paper 1, and I have a lot of things I want to revise, but before we get into any of that, I do want to make, I guess, like, a, a message. The last biology stream I did got 100 views, and I seriously, I can't thank you guys enough for that. That's amazing. But it does scare me a little bit, because that is a lot of people, and not a lot of you guys probably know, the deal with these streams. The deal with these streams is, I am an A-level student, I am doing the best I can to revise, and the best way that I find to revise is by explaining things to people. I am not a teacher, I am not qualified to explain anything. Please don't use these streams as a replacement for revision. Usually with things like the history streams that I do, I'll just send them to friends if we're doing an exam, and I think people might want to pick up on one or two things. I promise, I am not the best person in the world to watch for revision. It really does mean a lot that people are watching, but please don't depend on these streams for anything, because I can't promise that, I'll do everything right. So, with that out of the way, I also have a new tripod. It's cool, actually. You can, hold on. You can, like, raise it just by, like, this cog. That's cool. Anyways, let's get on with it then. Let's get on. I want to do some paper 1 revision. And I think we'll start off by going over digestion and absorption, because that is a topic that I haven't gone over in a good while. Maybe not since last year. And as a result, I want to make sure I know that. So, digestion and absorption. I think the hardest one is lipids, so we'll cover that first. Right. How does this one work? It is the emulsification of lipids by action of bile. And that would probably get you a mark in an exam, just saying how bile emulsifies fat or emulsifies lipids. I guess this could link quite easily to biological molecules. The structure of a lipid, that is going to be a fatty acid, and then, no, I'm completely mistaken here. Fatty acid, and it's the glycerol head, if I'm correct there. Emulsification of bile, the result of that is an increased surface area. Obviously, the purpose of increasing the surface area is so that enzymes are able to act more on the lipid, because a larger surface area increases the rate of enzyme activity. So, this will probably get you a mark or two, just talking about the emulsification of bile, increasing the surface area, emulsification of a lipid by bile. The bile does not get emulsified. So, the second point is, it is the action of, is it lipase enzyme? Lipase enzyme, which is going to break down our lipid into its core components, the glycerol head, the fatty acid chains. So that's kind of the digestion aspect done. The next thing is the absorption aspect, and this bit can get very convoluted very quickly. Right, what we have first of all is bile salts are going to associate with the fatty acids. And I think you're allowed to just say that in an exam. I don't think you need to go into detail. I think all you can really say is bile salts will associate with fatty acids of the glycerol. I like to say fatty acids because it just keeps me in check mode. This will form a micelle. Which is basically just like a vesicle. And the micelle will bring the fatty acids through the... Now, this is the annoying bit, because I don't remember what words you would use to describe it. It brings it through the cell surface membrane. How does it do that? I think it increases the solubility to lipids, so it makes these components more lipid soluble. Therefore, they are allowed to diffuse through the membrane. From there, they will combine, they will be reassembled by the Golgi. They will form chiromicrons, and they will then diffuse into the bloodstream via the release of, they will be placed in vesicles as chiromicrons, and they will be released into the bloodstream via exocytosis. That is a yap sesh and a half of a process. I highly doubt you could really drag this out any further. The most I would expect is maybe we get on paper 1 a 6-mark question talking about describe the process of digestion and absorption. But I feel like we're more likely going to get asked about the absorption, which is all of this process. I feel like you won't get asked to incorporate both. I feel like that's unrealistic. You would probably get asked a 4 or 3-mark question saying, describe a process of absorbing lipids into the bloodstream, or something like that. Absorbing fatty acids into the bloodstream. And then, this is the bit we need to focus on. Clearly, I need to work on the definitions, so let's go have a look. And then that should be digestion and absorption basically done. We'll do some exam questions, because digestion of carbohydrates is just breaking down, it would be breaking down things like sucrose into glucose and... Oh, I don't remember actually. Okay, maybe I do need to work on this. Let's have a look at the revision document. Okay. Digestion and absorption of lipids. Forgot to mention this occurs in the small intestine, but this was implied. Okay. Emulsification of lipids increases the surface area for faster hydrolysis. We mentioned this. Emulsification of lipids, increasing surface area for faster hydrolysis. We said surface area. We mentioned enzyme activity, which, yes, the enzymes will hydrolyze. So, yeah. Lipase action breaks lipids into component molecules, fatty acids. So we said this. Lipase action will break down the lipid into its core molecules, glycerol and fatty acids. Biosalts associate with the fatty acids to form micelles. We said this. Micelles bring the fatty acids to the epithelial cells. So, they bring, apparently this is all you need to say, they bring the fatty acids to the epithelial cell. Like, that's the word. I obviously never said that, but it's implied because I drew the ileum. Fatty acids absorbed by simple diffusion. Yep, because they're lipid-soluble small molecules. It's not facilitated diffusion. Although if you just put diffusers, that'll be fine. Fatty acids, triglycerides are reformed in the cell. and chiromicrons form as the triglycerides are modified by the Golgi. Vesicles move to the cell membrane via exocytosis. So, the only other mark point there, which probably would get you an extra mark or two, would be saying that triglycerides reform when they reach the epithelial cell, when they enter, and then the Golgi modifies them into chiromicrons. And that is the absorption and digestion of lipids. Well, okay. Very quickly, for those unaware, the absorption of carbohydrates and proteins is co-transport. That's why we learn about co-transport. Co-transport also occurs in the kidney nephron. But again, co-transport is one of those processes where if it comes up, we should all be thanking God, because co-transport is such an easy topic. It really isn't bad. So, digestion and absorption of proteins, we know this. Let's draw our polymer chain. Firstly, it's the endopeptases, which are going to break the interior bonds. So they break the bonds in the middle of the chain. Then our exo breaks the bonds at the end of the chains. And then our dipeptidizes, I don't know how you pronounce it, dipeptidize, I think. Yeah, they break it between the dipeptides. So very, very easy digestion. Just remember your peptidases. I don't know how you pronounce it. And then carbohydrate is something I don't recall. I think if we look at some exam questions, it might come back to me. Because it can't be that difficult. Exchange of substances. Mass transport, digestion, and absorption. Have we ever done paper on this? No. Here we go. Perfect. This is literally a 5-mark question. Describe the processes involved in the absorption and transport of digested lipid molecules from the ileum into the lymph vessels. Now, do we know how to answer this properly? If it's mentioning lymph vessels, let's just do this how I would do it. This is a 5-mark question. It's looking for 5 points, but for the sake of trying to make sure we get all the marks, we should definitely do 6 or 7. Right. Describe the processes involved in the absorption and transport. So we are at the part now, let's visualize this. We are at the part of a process now where we have our fatty acids that have been hydrolyzed. So, bile salts are going to... Remember, I'm not answering this properly, so I'm going to obviously use diagrams and stuff. Bile salts are going to associate with fatty acids, forming micelles. That should be a nice mark. Second mark, micelles bring the fatty acids to cell lining the epithelium of intestine. Third mark, fatty acids enter epithelial cell via simple diffusion. Fourth mark, we should probably kind of get a move on here. Fourth mark, triglycerides are reformed in epithelial cell. I'm just abbreviating it, don't do that in an exam. Triglycerides are reformed in an epithelial cell, and we'll also add to this, triglycerides are modified by Golgi body, Golgi apparatus. Finally, the whole bit of this is transport of the molecules from the ileum into lymph vessels. Finally, we will say they are placed in vesicles and they are released into bloodstream via exocytosis. That is, I hope, good enough. Let's have a look at the mark scheme. So, oh fuck, okay. I forget that mark schemes can be incredibly pretentious, and I forget how pretentious the digestion and absorption of lipids mark scheme is. You need to explain what my cells are for a mark. My cells contain bile salts, fatty acids, and glycerol. That would be a mark. They make fatty acids more soluble or bring the fatty acids to the cell lining the ileum. Why did I say epithelium? You know what, I probably wouldn't do that in an exam. Let's be generous, I'll give myself a mark. My cells contain bile salts and fatty acids / monoglyceride. Now I said bile salts associate with fatty acids forming my cells. I mentioned 2 components. I think I'll give myself a mark there, because I've listed the components regardless of context. Third mark, fatty acids are absorbed by diffusion, reject if by facilitated diffusion. Now I said they enter the cell via simple diffusion. That has to count. Surely? They are absorbed by diffusion, but that's a nice word. Instead of enter, they are absorbed. Fourth mark, triglycerides are reformed. Triglycerides reformed, that is perfect. That is a 1-for-1 copy of a mark scheme. Vesicles, you could also say I forgot chiromicrons. Oh my god. I completely forgot chiromicrons. I forgot to mention it, even though I just explained it. And finally, vesicles move to cell membrane, accept exocytosis. So, that is, overall, 5 marks. Some improvements we could have made is literally just making sure whenever they ask you a question about absorption, drill it into your head, you always use this same copy and paste paragraph. My cells contain bile salts, fatty acids, and glycerol. Instead of saying the cells lining the epithelium, always just say cells lining the ileum. That's what it's asking for in the question after all. Instead of saying that they move through the epithelial cell, say that they're absorbed. Mention chiromicrons. In this example, I still got the 5 marks, but it would have been a nice extra point. And finally, obviously exocytosis is that golden word that you can always use. So that right there was a very nice and lovely 5 marks. Could have improved it. I feel like in an actual exam I would have gotten 4 out of 5 for that, but I am more than happy, considering that is the first time I've answered a question like that in maybe 6 months. Next question. Figure 1 shows, it's this question, I've answered this before. Figure 1 shows a cell from the lining of the ileum specialized for absorption of products of digestion. SGLT1 is a carrier protein found in the cell surface membrane. It transports glucose and Na+. The action of carrier protein X is linked to a membrane-bound ATP hydrolase enzyme. Right, this is a difficult question, but let me demonstrate how this is meant to be answered, I think. Let me show you guys how I think I would answer it, and then, I don't know, you guys can think about this, because this is a weird question. Clearly, this is referring to co-transport. This is meant to be a glucose channel protein. This carrier X is a NAK pump, and this is meant to also be a glucose channel. And an Na+. So that's a symporter, that's a glucose channel, that's an NAK pump. Now, it's saying that this pump is linked to a membrane-bound ATP hydrolase. So, what is the purpose of the ATP hydrolase? This is to provide energy for active transport of Na+/K+ ions against their concentration gradients. That's how I think it should be answered. Let's have a look. Oh? There's a question missing. Okay. Releases energy. You cannot say produces, makes, creates. I said provides. I always get my wording wrong for ATP. You could have easily said ATP to ADP plus PI. That would be a mark. That is such a nice mark, and I missed out on it. Allow active transport of ions. I mentioned active transport. I mentioned ions. Energy allows ions to be moved against concentration gradient. So, I said provides energy. I feel like I would have gotten 2 marks for that, you know? I said it provides energy, but I didn't say how. And the how is it hydrolyzes ATP, ATP into ADP plus PI. That is such a nice mark, and I missed out on the most obvious part of it. The rest, I killed. I did that really well. I literally hit 3 mark points for the same mark point. But every other part there was slightly wrong. So I'm happy to give myself 2 marks for that. Well, 1 mark. It was a 2-mark question. I'll happily give myself 1 mark, but I know in an exam, it is always worth in an exam. That's something to note. It is always worth in an exam writing this or writing that. It is always worth it in an exam if you can do it. It's always worth it. In the context of oxidative phosphorylation, write this. In the context of even glycolysis, in the context of literally anything, write this equation because it could save your ass. It's like how they ask you to draw diagrams sometimes. There's a question here that it doesn't say the answer of. It doesn't give me the question, but it gives me the answer. And the answer is, structure is determined by relative position of amino acid groups, Primary structure is the sequence of amino acids. Secondary structure is formed by hydrogen bonds. Tertiary structure is formed by interactions in the R groups, creating an active site. Quaternary structure is more than one polypeptide chain. Now, I don't know what that question is, but if I had to guess, it's going to be something like, explain how amino acids are used to create an enzyme. And you would say the primary structure is due to the position of the amino acids. The secondary structure is interactions between the hydrogen bonds, forming an alpha helix, beta pleated sheet. Tertiary structure is due to the interactions between the R groups, things like disulfide bonds, hydrogen bonds, etc. Ionic bonds. The unique structure will create the active site. The movement of Na plus out of a cell allows the absorption of glucose in. This is active transport. I'm not going to answer this because it's easy. A concentration gradient of sodium ions, yep, because the sodium ions move out. Lower concentration gradient in the cell compared to the ileum. Sodium ions move in by facilitated diffusion. They bring glucose with it. Easy. Let's scroll down a little for some new questions. Complete the table by placing a tick in each box that you would expect to have shown a positive result. Oh, it's the whisking tube question. We've done this in a mock. And I already know the answer, but we'll do this again. So, it is literally... Also, after this, I probably am going to go, but I will probably come back later. We have a tube, and the tube is partially permeable. That means shit in the tube and shit out of the tube is going to mix. We have water. Water here. She carried out tests. Mixture of starch, suspension, and amylase. Okay. After 15 minutes, they remove the samples. So, firstly, the first test that they are asking is biurea. Second test is iodine. Third is Benedict's. And we need to say, will it be in the tube? And will it be in the solution? So, biurea test. Yes, there will be a positive in the biurea test, absolutely. Because biurea is the test for proteins, and amylase is a protein. Therefore, it's going to be in the tube. We'll come back to the solution in a sec. The iodine test, I don't know about this one, because how far has amylase got digesting the starch? If there's still some starch present, then I don't know. I don't know really. We'll take that for now. Then Benedict's is sugar. When starch is hydrolyzed, it becomes... I think this is the correct arrangement. Because it's saying it's partially permeable. Partially permeable. That implies that things like starch and amylase, big molecules, aren't going to be able to seep through, while things like sugar are. Am I correct in my assumption? I am correct in my assumption. Cool. That would be a question that would throw everyone off. Because everyone would assume that every biological molecule can diffuse through. Yeah. Absolutely. Absolutely. I want to look at quickly a question where it's asking you to evaluate. What can you conclude about the absorption? Here. What can you conclude about the absorption of a product of protein in the rabbit's food? This is question 4b. There is no significant difference because the standard deviations overlap. Therefore, okay, that is an important point and something that I want to talk about in a later exam. The idea of the statistical tests and the purpose of them. We have a bar chart and the standard deviations overlap. That means that there is no significant difference. Now, this question is out of 3 marks. You would get 2 marks just for saying that. So, error bars. Know about them. Error bars are significant. You could get 2 marks just for saying that there is no significant difference because the standard deviations overlap. Literally 2 marks. Now, if I was in an exam and I looked at this question, I would think, what can you conclude? What can you conclude? I would say something like, at 2.2, it's something percent. At 2.4, it's something percent. No. Talk about the overlap and standard deviation. And then you would need to describe that. You would need to explain why. The amount of protein in a diet is not a limiting factor. Explain the advantage of lipid droplet and micelle formation. Obviously, for this one, you would explain what a micelle is comprised of. You would say lipids, droplets, increase surface area for action of lipase. Micelles, you would say that they bring to the epithelial cell. That should be an easy question. Yeah, droplets increase surface area for faster hydrolysis. Micelles carry fatty acids through membrane. Cool. See, the thing is, once you understand how the mark schemes work, the questions become like see-through. They really do become see-through because it's a lot of repeating the same things. Cells lining the ileum of mammals absorb monosaccharide glucose by co-transport. Explain how. How many marks is that? That is 3 marks for explaining co-transport. That is lovely. I would not be complaining. And that is it for digestion and absorption questions. Okay. Honestly, I think that that's fair enough. Digestion and absorption is one of those topics where, like, a lot of it is common sense. You know, talking about, when starch is hydrolyzed, what happens to the water potential? You know, and then you have to say, starch is insoluble, but glucose isn't. Like, a lot of it is common sense. This is something that I need to drill in my head because I forget the importance of error bars. That is something that I do want to work on over the next couple of weeks as well, before paper one, is I want to go over the different question types. Where, you know the questions where it's saying, evaluate this, compare this. I don't know how to answer those questions very well, mainly because I suck at statistical testing, things like that. Another point is if they haven't done a stats test, then the data is bad. The reason it's bad is because then you can't tell if statistically significant. And finally is the p-value. I think it has to be over 0.00, it has to be under 0.005, I think. I think it's 0.005, let's have a look. Yes. It indicates statistical significance, meaning the observed data provides strong evidence against the null. That's obvious. You reject the null hypothesis if it's something like 0.038. If it's something like 0.85, it's not going to be statistically significant. It's likely you've obtained your data by chance. These are all of like the behemoths of stats and the idea that you need to drill in your head. If you get a question telling you, evaluate the claim that this medicine is important, look for error bars, look if they've done a stats test, look at the p-value. What other things? Look at the sample size. That is a huge one. And I don't just mean the overall amount of people. Like, if they've sampled 50 people, that's going to be way less reliable than, say, 100,000 people. The idea there is also species. If it's saying some shit like, this fertilizer claims to boost plant fruit production by 85%, I'm just making this up, but they've only tested it in strawberries, then you can say they've only tested one species. Therefore, you cannot argue that it will impact, every plant the same if they've only done one species. That goes for species in organisms. Obviously, I said organisms there, really. Also, the contributing factors. If it's doing a test on, let's say, like, age or something, it's in, basically the idea you want to get through is things that could make a difference, like age, sex, then there's a couple other generic ones as well. I guess like medical history. Just basic things like that.